Thank you so much for having me. I feel like this was actually a really good time to come in because we're going to follow up on this idea of like constraints of the system and what is it that ensures reproducibility. But we're going to go away from the very beautiful genetic details of Drosophila, and we're instead going to look at this amazing phenomenon of whole body regeneration. So the fundamental question, we'll see how much of a lag there is. Okay, the fundamental question that I'm interested in is regeneration, but specifically understanding morphogenetic properties and understanding how cells coordinate to rebuild and regenerate new organs. And so this is a question, this idea of organ morphogenesis is something that every animal has to undergo. Every animal that has tissues has to build them at least once during their life. And they do this normally through the process of development. And this is a crowd I don't really need to wax poetic about the beauties of morphogenesis too, but every animal will start with these beautiful cell divisions in the embryo. This is a C. elegans embryo I'm showing you here, and it will ultimately give rise to these really gorgeous structures, right? And so this idea of how do you go from a single cell to a really complex organ or structure or tissue or whatever it is that strikes your fancy is something that I think all of us are really interested in. But what the phenomenon that really brought me to my postdoc is this idea that while every animal has to do this once, there are many animals that actually have to rebuild their tissues. Sorry, there's like a really severe delay on the Zoom. There we go. There are many animals that have to be able to sort of recapitulate these morphogenetic processes and rebuild tissues over and over throughout their lifetime. And so we can, in reality, regeneration is probably more of a spectrum. We can largely break down animals that are capable of regeneration into those that are capable of what we call structural regeneration. So if part of the tissue is damaged or lost, they can rebuild it, but a large part of that organ still has to be present in order to give a template for stem cells to then replace missing parts. And then there's a second group of animals that are capable of this really amazing feat of whole body regeneration. So these animals that you see here on the screen, plus many others, all tend to live in the water. They're all marine invertebrates, and they're capable of essentially replacing any missing cell type after severe amputation, loss, or damage. And so this is something that many people have been interested in for a very long time. The earliest studies on Hydra were in the 1700s, where people were really trying to understand what is this amazing property and how does it work. And we've made a lot of progress at the molecular level in understanding how whole body regeneration works and what is sort of required for this process to function normally. And so you can kind of break whole body regeneration down into two main events. The first is that the animal has to sense and respond to injury. I don't know if this pointer will work. Sense and respond to injury. And we've made a lot of progress in animals like planarians and hydra and sea urchins and et cetera, studying gene expression, gene regulatory networks, a lot of this transcriptional events that we just learned somewhat about in the Drosophila. And we also know quite a bit about this idea of you have to sense what's missing and then you have to replace it, right? And so cells have to undergo proliferation, they have to make new cells, and then they have to sort of integrate into existing or non-existing tissues. And so in this area, we sort of have learned a lot about the positional cues that normally come from gradients of signaling activities saying, you know, make something here, don't make something here. But what we know very little about is the cellular level. And so understanding changes in cell morphology, how do cells navigate these really complex and variable environments to sort of rearrange and reform new structures. And so this is where I came in. This is why I joined Monsi's lab. And in order to ask these questions, you really need the right animal. And so ideally, you want something that checks all these boxes. You need something that's capable of whole body regeneration first and foremost, which sort of rules out a lot of the really beautiful animals that have nice tools, which is an ongoing struggle in the whole body regeneration field. You want I wanted something that was established enough to have some level of omic resources, right? Having a well-annotated genome is not something to knock. It's something a lot of us take for granted. I know I did when I worked in mice. A system where you can perturb the animal, whether that's chemically, genetically, physically. And then last, these two things sort of go together when thinking about ways to visualize cells. A lot of that comes down to having access to embryos. Oh, gotta annotate these in. And so this is what brought me to the animal I work on. Maybe. Sorry. The animal that I study is called Hofstenia miamia. It is a worm. It is called the three-banded panther worm as its sort of colloquial name, which it gets from this pigment pattern that's on the dorsal end. And so this is its head. This is its pointy tail. It's fairly straightforward, and it belongs to this really enigmatic phylum of animals. They are called acials. They belong to the phylum Xenocoelomorph. And my postdoc advisor was initially interested in studying acials because of this phylogenetic position. She's an evolutionary biologist. She was really trying to understand the evolution of regeneration. And in order to do that, one of the things that's really beneficial is looking at outgroups. And so acials belong to what is likely the sister outgroup to all other animals with bilateral symmetry. And so if you're trying to compare phenomena that you see in all of these phylums compared to cnidarians and ctenophores, acials provide a really opportune system with which to ask questions of evolution. But what worked out for, I think, the rest of the lab's advantage is that they're actually fairly easy to keep in the lab. So she collected these animals in Bermuda, and then you can study them both from the embryonic perspective. This is an embryo developing in the lab. It starts as a single cell and ends up sort of looking like a ball up until it looks like a worm. So it's not super interesting morphogenetically. And then these are just a video of little worms running around in a dish. And so we can culture them. We have thousands of these animals in the lab. And importantly, you can also isolate and inject these embryos to make transgenic lines, which I'll get to in just a minute. But the main reason why we are interested in Hofstenia is because it is capable of whole body regeneration. And so here I'm showing you, this is what one of these animals look like. If you cut it in half, the anterior fragment is going to rebuild a tail pretty quickly. And then the posterior fragment is going to have to rebuild a brand new head. And so this is what we call a blastema. It's a bunch of these stem cells have proliferated, and they're then going to differentiate into new structures. But as I mentioned before, the fact that we can collect these embryos in the lab means that we can manipulate them. And the easiest way, not easiest, but the most exciting way to do that is by making transgenic lines. And what this has done is it actually has allowed us to visualize cellular and tissue morphology at a really high resolution. And so here I'm showing you 3 different lines we have in the lab. This is an animal that has labeled the epidermis with an RFP. So you can see the anterior epidermis here, and then this is the pharynx, which I will talk about in detail in just a few slides. This is a transgenic line labeling the muscle, and this is labeling the syncytial gut. And then this was a video of muscle cells sort of assembling over the course of embryonic development. And so this has opened the door to a whole bunch of new questions that sort of move past the gene regulatory network level and start looking at actually cell and tissue dynamics. But one of the things that I think was most interesting about whole body regeneration isn't necessarily this, right? I just showed you that if you cut it in half, the head will reform a tail, the tail will reform a head. Actually, you can kind of do anything to it. So I've done all of these amputations in the lab, and no matter what the initial starting configuration is, it will always achieve the same end result. And so all of these organs will be formed, they will rescale properly. And so it sort of raises this question of tissue robustness in a way that I really wanted to explore. Yes?
What's the minimal that still grows back?
Yeah, I get asked this question every time. Theoretically, no one's done the experiment really robustly, so for that I can tell you. Theoretically, you need at least 1 stem cell to be able to make new cells. But what is the minimal positional information that's required for that stem cell to do its job? We don't know.
If you dissociate the animal to single cell, do they regenerate? Do the stem cells regenerate?
They do not form a perfect worm the same way that Hydra cells can reaggregate. I've done some preliminary experiments with that, and they do sort out to some extent. So I'll get epidermal cells on the outside, I'll get muscle on the inside that connects, and the thing will start beating. So it'll do some level of self-organization, but I don't know if the stem cells just didn't have what they needed at the time to build the right structures, or...
I guess clonal, I meant the state.
Oh, I see. Yeah. So the experiment, we isolate stem cells and transplant them?
You do like limit dilution, for instance.
Yeah. You could. I haven't done that. Yeah.
Like, can you spread the cells in a dish? So you have one, take your whole thing and...
Like flatten it or dissociate it and culture them?
Dissociate your animal.
Yeah.
And then flatten and spread everything out.
Yeah. You can. It will not reform a worm.
So it needs friends.
It needs friends, correct. The epidermal cells will kind of roll around a dish because they're superciliated, and so it's hard to get them to stay put. But they definitely need their interactions. Or like 1 single stem cell will not be able to make an animal. Does that answer your question?
So that makes his question even more relevant. What's the minimal amount of friends you need in order to be able to?
Yeah, the answer is I don't know. but I get asked it. It's a very tedious experiment to do. It's worth doing. I just haven't done it yet. Yeah. Okay. So the way that I chose to approach this question was by choosing this organ, which is the pharynx. You can maybe see the outside of the animal here. It's a little bit vague. This is a marker for the pharyngeal epithelium. And so the pharynx is a central epithelial tube in the animal. It's essential for eating. So food will go in through the mouth, into the pharynx, and into the blind gut. But it's also responsible for functions like mating and egg laying. So they'll actually lay eggs through their pharynx once they get mature. But most importantly, for my purposes, is you can amputate this animal and completely remove the pharynx, which basically forces it to build it from scratch. But one of the things that I want to keep coming back to during this talk is that the pharynx itself here, you can look at it from the single tissue level, and you have the pharyngeal epithelium. But in order to become functional, it actually needs to interact with all of these other cell layers, some of which interact functionally and some of which interact directly. And so the pharynx, as I mentioned, connects and feeds into this blind gut. But it's also wrapped by muscle, this really elaborate musculature system, and the outside of the animal is covered in a multiciliated epidermis, which will meet the pharynx at the top of the mouth and sort of provide this outer layer. And so if you were to take a cross-section through all of this and look at what this looks like at a cellular perspective, maybe, it'll look something like this, where you have the lumen in the middle of the pharynx, you have the outer pharyngeal epithelium, which will then connect with muscle, gut, and the outer epidermis. And so there is a luminal space in the middle of this tube. I don't know if this diagram is more helpful or not. I added this last minute, but essentially my question is, how do you rebuild an epithelial tube? And one of the things that is important to know is that all of these new structures during regeneration come from this population of pluripotent stem cells. And so they exist scattered throughout the parenchyma of this animal. And so essentially anywhere that there's not another cell type, you will find stem cells. So they're super densely packed. You see them most enriched here, but that's just simply because that's where there's the most space. If you cut off this fragment here, even though it looks like there's no stem cells there, it will still regenerate. So I think they just have lower PV expression.
Stem cell and germline.
At these stages, the animal has no discernible germline yet. They develop their reproductive structures about a month or two after hatching. They will come from the stem cells, yes. And so you'll start to see PV will be expressed in the ovaries. So they form ovaries on the ventral side of the body. How exactly the stem cells give rise to germline structures, we have no idea.
They can still regenerate once they are germlines.
Correct. Yeah. So they seem to form a second population of cells that is distinct transcriptionally and spatially. And so the reason why, one of the things that I was most interested in is that this question of like, how do you go from a stem cell to a differentiated cell type has been studied a lot in whole body regeneration, but it tends to focus on this aspect of it, which is that you start from a pluripotent state, you start either restricting or opening your chromatin, turning on gene expression, and you go to a differentiated state. And so there's a lot of papers out there about like, this transcription factor is responsible for this. But what I'm interested in is this idea that you're going from a population of cells that is extremely scattered. They're mesenchymal in morphology, so they have no known polarity. And then you have to sort of undergo this mesenchymal to epithelial transition and form this centralized and polarized epithelial tube. So this question is essentially, how is it that de novo pharynx regeneration is orchestrated? And this comes down to a question of tubulogenesis, which we know a lot about in developmental biology, right? Lots of people study tubes. Maybe my slide will show up. Lots of people study tubes. And the short version is that there's two different ways that you can form an epithelial tube. And the first of which is you start with an already polarized sheet of epithelial cells. Right, so these cells already have apical basal polarity, and you can undergo things like wrapping, where you sort of get constriction at this apical end. It'll hinge, and you can either bud this off to form a new lumen, so for example, in the neural tube, or you can start with something like this that just invaginates and ultimately forms something like this. So these are things that are extremely well characterized, both cellularly and molecularly, yes.
Instances where you just have a cell death from a solid block.
Yes. So the second option is essentially you have to make a new lumen. So rather than starting with an already polarized sheet of cells where you have sort of this inside-outside identity somewhat established, you have to make a lumen, which is the inside of the tube, from scratch. And so you can do that in a number of different ways. You can get sort of this aggregate of cells that will form a rosette, and then you'll start to, these cells will polarize and form a lumen in between. You can have something either cord or cell hollowing, where the same thing, you'll start making this empty space in between cells. Or, as you mentioned, sometimes that these cells that are on the inside will die off. And so when I was thinking about how this works in my system, I sort of came up with these two hypotheses that I would likely either see, this is a tail fragment where the head has been cut off and the pharynx is now being labeled in orange. I was like, I would predict either invagination of the anterior epidermis, which seemed like the simplest way to make the tube, or some level of de novo lumen formation, whether that's through forming a lumen and pumping it and inflating it, or whether it's about cells that are on the inside are dying. And then this transition from mesenchymal pluripotent stem cell to epithelial pharyngeal epithelium, in this case, would happen during this transition. When you start seeing a lumen, then you start seeing epithelialization. But what I started this characterization, and in this case, I'm showing you an in situ hybridization for the gene FAR. It's an unknown gene, but it marks the pharyngeal population really well. It's what I showed you as the marker for the pharynx in the earlier slides. Between 0 and 3 days, you see nothing. Once you start forming this dense aggregate of cells here, you see this aggregate of pharynx cells in the middle. It will become bigger. And then the states I'm going to come back to are a little bit messy, but it will always form an epithelial tube within 8 days. But when you start looking at these pharynges up close, what you might see is that there's a bunch of holes in the growing pharynx, right? Which kind of contradicts known mechanisms of lumen formation. You'd imagine that you need an intact sheet of epithelial cells if you're going to start expanding the extracellular space. If you have holes in it, it's going to kind of collapse, right? And so I took the approach of saying, well, this is an in situ marker. It's not perfect. It doesn't show you membranes. Maybe there are other cells that just don't make this gene. And so I started thinking of ways to visualize the pharyngeal epithelium through other ways. So as I said, this is the in situ. Using the transgenic line that we have, you can also visualize the pharynx here. And then there's an actin stain called suractin that works weirdly well on these fixed animals. Like, it works a lot better than phalloidin. And it stains the regenerating pharynx super strongly. And so I'm going to show you this real quick, but then we're going to come back to the actin stain. But what I want to sort of make clear is that when you start looking at epithelial cells, so this is now with the transgenic line, the only time you see the adoption of this epithelial cell morphology is after the tube has been completed. So at all of the intermediate stages, when you start seeing holes and all of these weird aggregations as the structure is forming, they're still mesenchymal. There's no signs of sort of epithelialization at the level of cell morphology. I've looked for the emergence of cilia. I've looked for sort of any sort of junctional boundaries. There's nothing I can see. This seems to be the clearest way of showing that it's like a switch that just happens. So I have tried looking for all your classical MET markers, PAR proteins, et cetera. None of them seem to be working super well in the system. So this transition is a little bit of a question mark still. And so I pivoted and started thinking about, oh, this is just a nice little, we're going to skip that. So I was trying to quantify this in any way I could. Any suggestions on quantifications would be deeply appreciated, because what I'm going to show you is that they're extremely variable. So if you track just the relative area and the size of these animals, it goes up pretty quickly. Within 10 days, it looks consistent with the areas that we see in uncut animals. But when you start looking at measurements of any sort of aspect ratio, here I'm showing you circularity, they're extremely variable. And this actually tracks with what we see with these intermediate stages. So here I'm showing you the actin stains. These animals were all picked. They were the same size. They were born on the same days, and I cut them, I anesthetized them, and made sure I cut them at the same place. And what you see is that all of these look very different. So this is the actin stain. I have pseudo-colored the pharynx cells just for the purposes of this talk, to try and make it really clear what I'm pointing out here. But hopefully what you can appreciate is that there does not seem to be a lot of consistency in what's happening in this trajectory. Yes?
So are the cells in these holes, right? Or is it?
There are, yes.
And so Are you looking at a population of cell that is proliferating and trying to find their way all together? Or are you looking at the cells in that mass already expressed that gives them
So-
the other ones you think might get it later?
Yes, I have slides on that in a few minutes, but the short version is all the proliferation is happening within the stem cell population. So once these cells have started forming these structures, they are no longer proliferating.
But you don't know where in these stainings, you don't know where the PV cells that are continuing to proliferate to
Yes,
build this mass.
yes. In these images, no. I can tell you based on other images that they are physically away from the center of the aggregate. Like if you were to do a transverse cut through it and look at it, you would see all these pharynx cells in the middle, and the stem cells are sort of further out, closer to the periphery of the animal. And I will show you what's on the inside of these, basically on the inside of, it's a good question though. I spent a long time trying to figure out what was going on. But one of the things, so I'm working with fixed animals here because the live imaging is quite tricky. Yes, Chandra.
It does appear as though the cells have a key problem they're trying to solve is just to line already existing gap. So there is already like
There's not a gap. I will show you, essentially what's on the inside is gut.
and near the holes, do you see increased cell division or anything of that sort?
There's no cell division in this aggregate. So what I did is because I'm working with fixed animals, I essentially had a sample size of about 800 worms that I fixed every 4 hours to try and get a better sense of what's happening temporally. And what I saw was that there seems to be this general morphogenetic trajectory. So this aggregation step is extremely consistent. It will always form right in the center of the blastema. The epithelialization in the final tube is extremely consistent. These intermediate stages, they will pass through this trajectory, but the timing of that varies by animal, and the exact sort of size of each of these pharynges is also quite different. So this is where it would be really nice to have a really reproducible system, but I'm working with what I have. So essentially the trajectory is that these cells will aggregate in the middle of the pharynx. At this point, the stem cells are around here proliferating. They will condense here. They will start growing along the anterior posterior axis as more cells are being added to this aggregate. And then they do this weird thing where they separate into two clusters, which I will get back to. It coincides with this opening of the anterior epidermis. And then the two clusters then start to sort of form these little bridges between each other, which suggests that they're maybe migrating towards each other. They do express, I don't have the slides in here, but they do express beta-1 integrin. I do think there's active migration between these clusters. And this is happening in a 3D sense. So I'm showing you the 2D sagittal view, but it's actually happening, this convergence is happening in a circle. And so they're forming a tube. And ultimately they will end in this epithelial tube.
Is derived from one single stem cell or?
We have no way of knowing. That sort of like clonality of lineage tracing, we don't.
So you don't know whether there is replacement during this process or whether this is all derived from this tiny primordium?
Oh, I see. I think right now what's happening is that you need constant recruitment of more stem cells. Yeah. So I do have some slides on that in just a minute. Yes.
You mentioned that it's not reproducible, but it's a fundamental principle of regeneration. The end structure has to be the same, but the intermediate could be different.
Yeah.
So the question is how does it take place during development?
It's a really good question. I have started looking at that. And the short version is it seems to form totally differently. It seems to form just through invagination of the anterior end, which is interesting in and of itself to try and understand why is it different and how is it different. Are the intermediate progenitors different? I have no idea. It's one of the things I would like to do when I start a lab. Okay, so this was my best attempt at trying to quantify this. This is a percentage of total animals. This is about a sample size of about 500. Hopefully what you can see is that 100% of cells start here, 100% of animals end here, and all the intermediates are a bit messy. So I have decided to break this down in sort of a temporal step and trying to explain one step at a time. And maybe gain some resolution as to what's driving each of these transitions. So the first thing that I was trying to understand is this growth, right? And this idea of how is it that the pharynx is getting bigger? And it could be that the cells themselves are proliferating, right? I was trying to rule out these different options. Is there proliferation within this population of cells? Are you getting induction of neighboring cells and there's some sort of fate switching, or is it that more cells are being recruited from the stem cells? Yes.
But then in between the steps, do they start over from aggregation step or?
I've never done that. That's a good experiment. Like halfway through this progression, like get rid of it entirely or get rid of some of it?
Somewhere in it, but it's somewhere in between. See, they're trying to start from aggregate or continue.
Yeah, that's a good question. So this is something that I've spoken actually with Maha quite a bit about, and our best guess as to how this is happening is that you have essentially constant recruitment of new progeny throughout this whole process. And so what signals exactly are coming from those cells to recruit them and do a directed migration, I'm still trying to figure out. I have a little bit of data on that. But my guess is that if there's, I don't think it's in this talk. Essentially, one of the comparisons I did was I cut the animal through the pharynx and left part of it behind, whether that's the anterior or the posterior part, and you get none of this. The cells just get added and it just gets bigger. And so I think there's probably some level of communication from existing pharynx cells to new pharynx cells that they're just getting recruited and adding into an existing structure. It's whenever you get rid of it entirely and it has to sort of self-organize that it goes through this trajectory. So my guess is that if I were to do that, it would just get bigger and follow up on where this is, but I don't actually know. It's a good example. Okay. So the first thing I looked for was this proliferation idea. And essentially what maybe you can see, I have quantifications that are not on this presentation, but there's very little proliferation actually within the pharynx itself. It's all happening in these stem cell populations that exist either within the new tissue here, this is at 3 days, 4 days, this is 4 days, versus whenever the anterior end starts growing a little bit more, it sort of gets more restricted to this mid-body approach. I'm doing EDU tracing to try and see if there's like anterior posterior differences and where new cells are being added, but I don't know the answer yet. And so this was not entirely surprising. This was the dogma in the field that all the proliferation existed within the stem cells. But now I was trying to sort of understand this difference between, is it that neighboring cells are being sort of induced to join, or is it that new cells are getting recruited? And so to do this, I had to take a sequencing-based approach and ask this question of, is there an intermediate population of cells? So when you look at existing sequencing data sets that we have, cells either express PWE or they express this pharynx marker, but they don't express both, which was suggesting to us that this is the more differentiated or final form of the pharynx cells. And maybe if we see this intermediate population, we can start to get an understanding of maybe are they right next to the pharynx cells or are they coming from further away? So this is a subsetted UMAP. So I took all of the data sets and pulled out cells that either express PWE or that express FAR, and they don't express both. And then I looked for genes genes that were sort of co-expressed in both of these clusters. And there's a whole host of genes that you can sort of subcluster them out. But one of the things that I found was this gene FB1, which if anyone knows about it, it's sort of involved in axon guidance and directed cell migration. And when you do an in situ hybridization for it, hopefully what you see is that they're kind of scattered all over and they're also joining the pharynx here. And so they seem to have an intermediate distribution. A couple of the other in situs for other genes have similar distributions. So I think what's happening is that there are pluripotent stem cells that are sort of becoming specified to different cell types, and then they're actively migrating and joining this pharynx. And so what I did is I knocked down this gene to try and see what happens. And it prevents proper pharynx regeneration, which is not entirely surprising. Some of them do make it, but a lot of them kind of get stuck in this intermediate phase and they can't fully epithelialize. But the biggest difference is that the overall pharynx is smaller, which suggests that fewer cells are being added to the pharynx as it's growing, which would sort of support this idea of directed cell migration. But it is just a hypothesis at the moment. Yes.
But do you see that population of intermediate genes if you don't perform one of the cuts?
No. There's a few cells, but they don't seem to be seen in the pharynx. It seems to be a regeneration sort of specific population. Yeah.
other events of regeneration taking place in this mutant? Or
Is what? Sorry?
are other event of regeneration taking place in this mutant? So if you look at other structure that
Yes.
must regenerate.
Yes. I have looked at, they form mouths. So the epidermis seems to be fine. They form neurons. And so they seem to be forming the brain at the anterior end. So it does seem to be somewhat pharynx specific, but I haven't done the extensive cataloging. I need more and more animals to do that. But it's a good question to see how specific it is to this tissue. Was that your question?
Yes.
Yeah.
So my question is that if this gene is only involved in migration, you would expect to see the pharyngeal cell differentiating close to the kiwi.
I see.
You don't. So
yeah.
So I think it's the role of this gene is not only to, or I mean, the mechanism must be that the cells start to differentiate when they reach. I mean, you could explain your growing structure and that it only grows at a given place.
Yes, I think it's probably Wnt signaling dependent. I also think that my guess is that there's some level of contact dependence because all of the pharynx positive cells are always in contact with another one. And so I think there's probably something else that's inducing the final differentiation. It's really hard to untangle that, but I'm trying. Okay. So my conclusion off of this preliminary data is that the expansion of this growing pharynx is likely due to recruitment of stem cell progeny. But the thing that I was really trying to figure out was this strange morphogenetic trajectory. And so trying to get some level of understanding of what is driving the organization of these clusters and maybe what is constraining one movement versus the other. And one of the things that kept showing up in the sequencing data we had were genes that were related to MEK-ERK signaling. And this is not entirely surprising because MEK and ERK are involved in tissue organization in like every system, right? And so thinking about this trajectory is you have receptor ligand binding at the membrane that will then sort of set off this kinase cascade, phosphorylating the MEK kinase, which will then phosphorylate ERK, which will then turn on all sorts of genes that are associated with every cellular process imaginable. And so the first thing that I did was I just stained for phospho-ERK in this animal. And what I saw was that essentially only these intermediate stages have phospho-ERK expression, and it's super high, and then it goes away entirely. And so what this was suggesting to me, and then the interesting thing also is that it doesn't really seem to be in the pharynx itself, it's in the neighboring cells, which became interesting for a couple of different reasons, but I wanted to know if this was functionally relevant, and so I used a MEK inhibitor, specifically at these temporal time points, specifically to try and understand if I block MEK-ERK signaling, what happens to these pharynges. And what I see is a really clear phenotype where they essentially cannot converge to form this tube. And so all of these animals sort of get stuck at these intermediate stages, and they almost start to form little mini clusters. And some of these start to look like they're trying to epithelialize with each other, but they can't quite get there. And so if you leave them in this long enough, they'll die. But if you do this 48-hour window, either earlier or after, you do not get the same phenotype. And so something about it is specific to sort of the convergence of these cells towards each other. Right now, what I'm trying to figure out is essentially whether or not that is due to the muscle cells that are surrounding the pharynx clusters, and if there's something going on there. There's a whole contractility thing. But what this data shows me, at least, is that something about MEK-ERK signaling is required for the convergence of these clusters. What it doesn't tell me is what's happening here. And so this was, I didn't really believe this data for a very long time. I was like, oh, they're getting squished or something is happening, but then I kept seeing it over and over and over again. And one of the things that I was trying to understand is whether or not these cells were migrating laterally, which happens in, I think, tunicate heart development, or whether or not they're being physically displaced. And as I mentioned earlier, the new pharynx has to functionally integrate with neighboring tissues, right? It has to contact the epidermis to form this mouth, and it has to sort of form this sphincter. The sphincter at the posterior end, which leads into the gut. And so one of the questions that I started with was just trying to ask, how are the epidermis and the gut being remodeled during this transition, right? And does that give us any clues as to what's happening? And I pointed this out a little bit, but what you start to see when you zoom out and look at this transition is that the anterior epidermis actually undergoes some quite significant changes that correlate almost perfectly with what's happening with the pharynx. And so the first thing that we start to see is this little dimpling of the anterior end. This will invaginate and it'll get deeper as the pharynx grows in size, and then it literally just like rips open. These ripped epidermal edges will get brought close together whenever the pharynx converges, and then ultimately they will connect to form the mouth. And so this is just a very crude video of a tail fragment that hopefully you can see the invagination happening at this anterior end, and it will just start contracting and compressing, and all of these cilia are here. So this anterior end is still closed, but this is what precedes that. And so right now what I'm trying to do is, I don't know why it's so laggy, but I'm trying to figure out a way to quantify sort of the compression of this animal as it's doing this and correlate that and try to visualize the exact moment where it separates. If anyone has ideas as to what is happening, I would love to chat, because right now I can describe it and I cannot explain it. But what I do know is that the anterior opening that I see correlates perfectly with the separation between these pharynx clusters. And so I do think something about this separation is mechanically linked. So whenever they're opening this way, whether or not the anterior epidermis opening is what's physically splitting the clusters, or if something else is happening there, if you plot them against each other, it's like an extremely close correlation. Yes?
It's an opening. Could it be that the cells that were on top are just migrating in?
It could be. I've been saying opening because we see cells leaking out. So it is
Out.
Oh yeah. Give me the next slide. So the second question basically that I was looking at the same time is what's happening with the gut.
Oh.
Yeah. And so the other question is this gut. And these animals have a syncytial gut, which is quite strange, and we don't understand fully how it works, but it seems to function like a fluid-filled bag, and it's just going to take up space. And so it's surrounding the pharynx as these sort of these early stages are happening. And then at the point of this separation, we start to see gut coming out of this opening. Whether or not the gut is pressurized and is actually pushing through the epidermis, I'm trying to work with Maha and figure out how to test this. But one second. But what seems to happen is that as this closes back up, we see, no, I guess this is the next slide, but you can see this even in animals that have been fixed and have not been stained. Oh, I don't know what happened to it. Okay, it was just a picture of like an animal that has, okay. Anyway, you had a question.
Yes. So when you look at this stage of opening, and specifically at the epithelial cells in that anterior portion, I'm not sure how frequently can you see this restitution of epithelial cells described in some acils. They do get sunk in. Do you see that happening more here during this stage, or do you see cilia in these epithelial cells in that portion still being intact? Like, do these cells look normal, basically, as the gut is kind of protruding there?
It does not seem like it's an intact epithelium that has now sort of moved laterally or sideways.
Those epithelial cells that are neighboring the gut, just at the edges of the hole, do they still look like normal epithelial cells when you?
It is very hard to see the exact morphology of them, so I wish I could give you a really convincing answer. I don't know. It looks like I have a separate project on wound healing, and they look very similar to that, which is why I've been using the like opening, because it literally looks like an injury.
Yeah.
And so I have been trying to figure out what's causing this opening and what causes because eventually the pharyngeal epithelium has to contact the outer epidermis. And it was strange to me that this was happening, that these events were not linked, because in normal mouth formation, like that is sort of what leads to then the dissolution of the stomatia collar and all of that. And these don't seem to be happening at the same time. So I think the consequence of this opening happening before these epithelial layers have contacted each other is that you get this gut leaking out. Yes.
Again, is it a regenerated gut that insert in, or is it the whole gut?
Because it's syncytial, we have no way of knowing what's new versus old. The tail fragment has pre-existing gut.
Yeah.
What seems to be happening is that new cells, I don't know if new cells are getting added or if there's like ion channel calibration, because you do see the gut sort of like inflate. The animals sort of become like little balloons. So I think something is happening within the gut, but we would have to do some level of like nuclear labeling to see where new cells are sort of getting integrated into the gut.
Because I don't understand how you regenerate when you showed us the quarter part, where you showed that you had just the anterior part. There is no gut there.
Yeah.
So
So there is pretty much anywhere you cut it. So even if you cut through this here, there's a little bit of gut on either side. And so it becomes so it goes up on either side of the pharynx. And so a lot of this animal is gut. It would be interesting to try and get rid of the gut entirely and see what happens. I really want to figure out how they're fusing.
The ejected gut doesn't give rise to a new animal.
Yeah.
Would be pretty side.
Yeah, I don't know. Because there's cool, like you could imagine a line where you try to ablate all these cells or something and see what happens. People have done that with neurons, but not really. No one's interested in the gut. But what I see quite consistently is that as these cells sort of converge around, you get what almost looks like compression of the gut. But this is what's filling those holes, is it's just gut. And then as the pharynx forms and as it connects to the anterior end, this little packet of gut just gets expelled out. So it's a very strange solution to a complex 3D problem that I think maybe would only work in this animal because of the syncytium, but it seems to work very, very well. Okay, so I have not actually answered whether or not this is driving this process. What I can say is that it's somehow coordinated with, likely involved in the separation of these clusters. I'm trying a couple of crazy experiments, like trying to inflate the gut or deflate the gut and see if changing the pressure changes what's happening with these cells, if they get displaced at all. But those are all still works in progress.
No idea is that I was going to say GF, but it's not a this signal, that this ERK signal comes from the gut or from the.
I think the ERK signaling is coming from the muscle, which I didn't really talk about, because there's also muscle that sort of connects the pharynx to the outside, and all of this invagination is muscle dependent. So if you anesthetize the animals and you block muscle contraction, they can't do any of this, and they can't do any of this. And so I think there's some level of contractility that's associated with this, but trying to figure out exactly which steps are driven by the muscle contraction has been a bit of a challenge. So that's what I'm doing right now. But yeah, I think that I've been trying to find the ligand for the ERK signaling. They do have an FGF. The FGF inhibitor just kind of kills them, which is not helpful. So I've been trying to figure out other ways of getting at that. I want to do a lot more temporal sequencing now that I have a better sense of what's happening. So trying to separate out the animals by morphogenetic stages rather than time points and get a clearer representation of what each of the sequencing landscapes is happening, each of the transcriptional landscapes. Okay, so the summary of where I'm at right now, this, as I mentioned, this is a bit of a work in progress, is that the regeneration of this pharynx sort of requires integration with other cell types. So you can't actually look at it at just trying to figure out what's going on within the cell population, because you need recruitment of new cells from the stem cell pool. If you get rid of the stem cells, none of this happens. You need remodeling of pre-existing tissues to sort of create these spatial boundaries. And you also need some level of cell-cell signaling to direct either the positioning or sort of the interaction of cells with each other. And so my hypothesis is that these tissue level mechanisms are actually what's responsible for buffering this immense cellular variability. And so if you can imagine a world in which, because you're getting more cells from somewhere else, it's sort of ensuring that you always have enough cell numbers. If you have an animal that starts with fewer cells than the other, maybe you get more proliferation to boost up and make sure that you have the numbers of cells that you need in order to make a properly sized pharynx. As I mentioned, sort of this remodeling of other tissues creates the spatial context for the tube to form. And then the signaling between cells is what's providing sort of the cues for one cell to go somewhere else and for these clusters to now interact with each other. And so I think this is really exciting because what it really tells you is that this phenomenon of whole body regeneration is super imprecise. Like we're talking about imprecision at like the transcriptional level, and it's super minute. This is extremely messy. And it really requires interaction and coordination between cells to compensate for the extreme variability between animals. And so if it required cells to be in the right place with the right neighbors, expressing the right genes at the right time, this process would fail most of the time, right? And instead you have a whole bunch of animals who have been able to sort of compensate and sort of work with extremely variable starting conditions and always get to the same endpoint. And so I think that robustness is interesting enough to overcome the reproducibility challenges and a lot of the complex technical details that are in the intermediate stages. But a lot of this is still work that I've got going on and trying to sort of mediate intermediate steps. So any feedback would be much appreciated. But for now, I'd like to thank my lab. Monsi has been extremely welcoming of me coming in and trying really crazy things. I had lots of help from Scrub and OEB and the grants office, and thank you to my funding. Thank you to all of you for your attention, and I'm happy to take questions.